Hi everyone. Today I'm presenting my work on residual embedding similarity-based network selection for predicting brain network evolution trajectory from a single observation. This work has been done at Basira Lab by myself, Ahmet Serkan Goktas, and supervised by Ela Besedok and Dr. Islam Rekik. This paper is accepted to Prime MICCAI 2020. Longitudinal neuroimaging of the brain has spanned several neuroscientific works to examine early disease progression and improve neurological disorder diagnosis. Existing brain evolution trajectory prediction studies mainly focused on Euclidean neuroimaging data such as magnetic resonance imaging (MRI) from a single baseline time point, such as predicting the 6 months or 12 months follow-up MRI of a given subject. However, such frameworks were designed for brain images, so they might fail to generalize to non-Euclidean data, such as brain networks, which mainly reflect the connectivity patterns existing among brain regions. So, the question we aim to answer in our work is, how can we design a framework for a brain network time-dependent evolution trajectory prediction? Previous methods are mainly focused on using MRI images as their source of input. This is insufficient since the interconnectivity of the brain cannot be exploited compared to using brain networks. There is one work that uses brain networks. However, it uses raw feature vectors which fails to preserve graph topology. Also, it does not take the population traits into account, which means that shared traits between subjects are overlooked. To overcome these limitations, we propose residual embedding similarity-based network selection or RESNets. This framework can be split up into 3 steps. At the first step, we perform brain network embedding at baseline time point using adversarial connectome embedding. At the second step, with the help of netnorm, we produce the CBT or population mean denoted by X sub C. Using this CBT, we perform CBT-based embedded residual construction. At the third step, using the residuals, we predict the brain network evolution trajectory for the testing subject. Basically, using adversarial connectome embedding, or ACE, we come up with embedded vectors of brain networks. The reason for that is we want to embed the graph topology to a representative vector instead of using feature vectors. The representativeness comes from the way unit architecture of ACE is trained. Essentially, unit trace to minimize the error between reconstructed or X-hat network and original network. In a nutshell, we have embedded vectors of each subject and we will use them instead of feature vectors. Our notion for similarity detection of the subjects is that an individual sample can be thought of as a deviation from population mean. Therefore, if we compute how each subject deviates from the population mean, we can compute the similarity based on this information. In order to come up with a population mean, we propose to consider the inherently shared connectivity patterns which are captured in an average population network called connectional brain template, or the CBT. To this aim, we leverage netnorm, which estimates a normalized connectional map of a population of brain networks. Feeding the training subjects at baseline to netnorm, we come up with our CBT, or population mean. To use the CBT for producing embedding residuals, we feed the testing subject and CBT into ACE and embed them as it was done in the previous step. To compute the embedded residuals, we use the absolute differences of embedded CBT and subjects, and we finally have our embedded residuals. At this step, we calculate the similarity between embedded residuals of each subject and the testing subject. For example, we compute the cosine between vector R1 and Ri and put the result in the score vector. Similarly, we repeat this for each vector Rn and Ri. The lower the cosine, the higher the similarity. Then, for each time point, we average brain networks of selected subjects and come up with our prediction. Finally, we have our predicted brain network trajectory of the testing subject for each time point. For testing our network, we prepared 67 subjects from Ethnigo public dataset. For the weights of the edges of the brain networks, we used absolute difference of mean cortical thickness between regions of interest. For evaluation metrics, we used mean squared error and mean absolute distance. We used leave one out method as a cross-validation strategy and varied the value of k, which is the number of selected subjects, between 2 and 4. We benchmarked RESNets against three frameworks. In SNets, the similarity between subjects are calculated by dot products between raw feature vectors. ESNet does the same thing with embedded vectors. As we can see here, ResNets outperform other frameworks at both of the evaluation metrics. As a summary, our work is the first work on brain evolution trajectory that uses UNets. Our work also uses CBTs that leads ResNets to outperform other methods. Our future work includes extending the framework to handle multi-view datasets and implement joint embedding using ACE. For more, you can visit basura-lab.com. Don't forget to subscribe, like, share, and cite our work. Thank you for watching.